In this video, we're going to focus on the plasma membrane, the structure and the function, and why it is important in a cell. We'll come back to all those other organelles in the next lecture video. Now, I would argue that in a eukaryotic cell, the two most important organelles are the nucleus, which contains our DNA, and the plasma membrane. Now, you can sort of think of a loose analogy for the plasma membrane as like the walls of your house. The walls of your house provide support, they provide protection, and the doors and windows of your house allow for things, including people, to move in and out. Now, that's a really loose analogy to our plasma membrane, but the plasma membrane does hold the contents of the cell and regulate what moves in and out. So that's sort of how you can think of that plasma membrane. There are some major differences in that analogy. Major problems in that analogy. The big one being that the plasma membrane in a cell is soft and fluid, not a hard, rigid structure like your walls. You could also think of it as kind of like a Ziploc bag, maybe, just some sort of material that holds all of those contents. But every cell of every living organism is enclosed by a plasma membrane that regulates its interaction with the environment. That plasma membrane is made up of two layers, and you can see those two layers in this picture here. If you look, it's hard to draw, but there's two layers there. So the outside membrane of all of your cells is a bilayer or two-layered. Now this is a little different than some of the organelles inside the cell that are actually only a single layer membrane and may have a slightly different composition, but all the membranes in the cells are going to be very similar to the plasma membrane. Main functions of the plasma membrane are to hold contents of the cell in place. So we've got our plasma membrane and we've got our cytoplasm, which is that fluid jelly-like structure, jelly-like fluid inside the cell that suspends all of those organelles. Now, outside of the cell is extracellular fluid. So this is usually a watery fluid that flows, washes over all the cells in an organism. And then inside the cell is the intracellular fluid. So the cytoplasm is the actual jelly-like fluid. The intracellular fluid contains the cytoplasm and all the organelles that are found within the cell. The plasma membrane also works to take in food and nutrients from the environment. We'll talk about how it actually does that in a few minutes. Basically, it will engulf or surround food particles and bring them into the cell. But there are several ways that cells can bring in nutrients from outside of the cell. The plasma membrane also helps to build and export molecules. So a lot of our cells have the function of making products that are important in our body. Things like hormones, things like fats, sugars for storage, proteins. All of those products that a cell is making has to be exported if it's part of a multicellular organism to be used in different locations. And then finally, the plasma membrane allows interaction with the environment and the neighboring cells. So it not only encloses the cell, but also protects it and allows for communication and exchange of products amongst the greater environment inside an organism or with neighboring cells. The plasma membrane that surrounds the outside of all of our cells is called a phospholipid bilayer. So let's break this down and see what on earth all these words mean. Phospho means phosphate or phosphorus. Now, we skipped the two chemistry chapters in our textbooks, chapter 2 and chapter 3, because at SWIC we feel like you don't necessarily need all of that chemistry to understand the biology. But the chemical makeup of all of our cells is really important, and it is likewise important in our phospholipid bilayer. So the phospholipid bilayer is made up of a phosphate group bound to a fat or lipid group. Okay, so you actually can see that here. You'll have to take my word for it. This is your phosphate group on top, and this is your fatty acid or lipid group. And then bilayer refers to the overall structure. So the phospholipid part are the individual pieces, and the bilayer part refers to the way that those pieces are arranged. And if you look, they're arranged in 2 layers. So we have a 2-layer structure that is made up of these phosphate and lipid bilayers. So let's talk about the characteristics of the phospholipid. So this structure here is called a phospholipid. Again, because we've got the phosphate group on the top, fatty acid lipid on the bottom. Again, lipid is just another word for fat. Now, because of some chemical properties that we won't go into here, the phosphate group is attracted to water. So, a couple of vocabulary words for you here. We say that something that is attracted to water is hydrophilic. Hydro meaning water, philic meaning liking. Okay, so a water-liking molecule, a molecule that is attracted to water. And we also say that that phospholipid molecule is polar. Now, polar means that it has a slight positive or negative charge. Now, that polar positive or negative charge allows it, or what causes it to be hydrophilic. And when this hydrophilic head is attracted to water, that's really important because it is going to face all the water inside and outside of our cells, which is why you see these phosphate heads all lined up to the interior or exterior of the cell. The fatty acid tails, on the other hand, have the opposite properties. They are hydrophobic and nonpolar. So you can probably guess what hydrophobic means. Hydrophobic actually means water resisting. So if you have a phobia against something, you don't like it, you want to stay away from it. So water resisting, it wants to stay away from water. And the reason for this is that it is nonpolar, which means it does not have a charge. So it has no charge. Water is actually charged, which is why polar molecules are attracted to it. Things that are not charged are repelled by water. So nonpolar molecules are repelled by water. The reason for the phospholipid bilayer has to do with this polar and nonpolar structures on the phospholipids. So I'm actually going to clear all of this really quickly. If you'll look, as I said a minute ago, we've got the polar hydrophilic region oriented out and in, and that's because that's where all the water is. We've got water, or that extracellular fluid, on the outside, and that intracellular fluid that's mostly water on the inside. So the hydrophilic polar sides of those phospholipids are going to orient towards the inside, and the non-polar sides are going to be attracted to one another, repelled by water, and face toward the inside. Now, all of these little phospholipids can shift and sort of move and squirm around within their current plane, but they cannot flip. They cannot turn and face the other way because of those hydrophilic and hydrophobic properties. So these membranes stay together based on these chemical attractions or molecular attractions to keep the shape of the phospholipid bilayer. Now, this structure, this hydrophobic and hydrophilic structure, is also key in regulating what goes in and what comes out of a cell. So here we have sort of a side view of our plasma membrane. And again, you'll see that we've got our hydrophilic heads on the outside and our hydrophobic tails on the inside. Now this is important because that means any molecule that is itself hydrophobic is going to try to get in and it's going to be repelled. It's not going to be able to just push its way through. But it also means that any molecule that is hydrophilic might try to get through, but it's going to be repelled by the hydrophobic tails. And so this keeps things from just coming in willy-nilly, moving out all on its own. Okay, remember that one of the main functions of the plasma membrane is to control what goes in and out of the cell. But things do need to move in and out of the cell. Oxygen needs to move in, carbon dioxide needs to move out. Sugar molecules need to move in. Hormones or other products need to move out. Water needs to move in and out. All of these molecules need to move in and out of the cell. So we do that by the presence of transmembrane proteins. So we're going to focus on these proteins, but you do need to know the general properties of these other ones. But transmembrane proteins are going to regulate and move things, different kind of things, in or out of the cell. Now, the reason that they can stick inside the plasma membrane is that they too have hydrophilic and hydrophobic regions. So those hydrophilic regions are going to be attracted to the hydrophilic region of the plasma membrane, and they can get stuck in there, stay in the membrane, and perform their jobs. All right, so there are many molecules that perform important functions in the plasma membrane. I would like you guys to be able to identify these and know the general sort of the general function of them, but we are going to focus mostly on those proteins that provide transport in and out. So we have something called a glycoprotein. Glyco meaning sugar, protein meaning protein. So this is a molecule made up of sugar and proteins, and they actually act as a fingerprint or identification molecules for the cell. So other cells can recognize what they are. We also have glycolipids. These are carbohydrates attached to a fat. Then we have cholesterol. Cholesterol is really important because it helps the membrane to maintain its flexibility and fluidity. So the amount of cholesterol within a cell membrane is going to regulate how much these little phospholipids move around. If a cell gets too cold, they're going to sort of seize up a little bit. Think about when you put butter or oil in the refrigerator. If it gets too hot, it becomes more fluid. Think about if you leave butter on the counter on a hot day or put butter in a pan, it spreads out and essentially melts. So cholesterol helps to regulate that fluidity. You don't have to know how it does that. I just want you to know that it does that. All right, we are going to focus though on the proteins that help move things in and out. These are transport proteins because they provide a passageway for molecules through the cell. There are some other important proteins. Receptor proteins bind particular molecules that allow for regulation within the cell. Recognition proteins, along with their counterparts, the glycoproteins, can help as identifiable fingerprints for the cells. Now, anything that's on the outside of a cell can allow another cell to recognize it. If you think about COVID-19, you probably know that it has a spike protein that binds to specific cells within our bodies, particularly our respiratory cells. Those spike proteins are recognizing some sort of protein or glycoprotein on those cells to know that that's the kind that it infects. So those receptor proteins and recognition proteins are really important. And then we also have membrane proteins. These guys are proteins that help to facilitate chemical reactions in our cells. We're not going to focus on those too much either. But we are going to focus on these transport proteins. We'll get to those in a minute. But let's first talk about why membranes are so important. So we've already talked about a couple of things. They provide structure for the cell. They provide an outer boundary for the cell. They regulate what comes in and out. We'll talk about how they do that in a minute. But there are some other reasons why membranes are important. One is that faulty membranes can cause disease. So here we have some photos of individuals that have cystic fibrosis. So cystic fibrosis is caused by an incorrectly shaped transport protein, like this guy here, an incorrectly shaped transport protein that comes from a mutation in our DNA. So the DNA is incorrectly coded to produce a faulty protein. Now that protein cannot be embedded in the membrane correctly because its hydrophobic and hydrophilic areas are essentially mixed up. So it cannot stay embedded within the membrane. When that happens, it pops out. Without that membrane, chloride ions cannot get into the cell. They can't be transported across the membrane properly. Those chloride ions cause a buildup that produces a sticky mucus, thick sticky mucus, that lines the internal organs, including the lungs of the individual with cystic fibrosis. So basically, their lungs are lined with a thick, sticky mucus that prevents the lungs from correctly bringing oxygen into and out of the cell. So there are lots of other examples of cellular diseases. We can't go through all of them there. But this is one example of how a faulty membrane can cause disease. So we need proper structure in our membranes so that our cells can perform proper functions. Another reason that they're important is that pharmaceutical drugs can alter membrane functions. So remember we said that there are receptor proteins that identify molecules and bring them into the cells. So an example here are beta blockers. So normally we have adrenaline molecules, it's our fight or flight molecule, that binds to specific receptors on the cell, and that then starts a cascade of reactions that causes our fight or flight, our increased breathing rate, our jumpiness, sort of our, whether you are going to stand and fight or whether you're going to run away, that response. But beta blockers are a type of pharmaceutical that actually bind at those receptor proteins instead of adrenaline so that it reduces the reaction to a stressful situation. So essentially reducing anxiety and stress in normal everyday situations. So our membranes are important because they are taking chemical signals from the environment, from our bodies themselves, and using them to create a particular cellular response. But we can also use drugs, prescription or non-prescription, to block those receptors and change the reactions of our cells. So membrane surfaces can also act as a fingerprint that identifies cells. Now this example here is actually HIV, but if you've watched the news in the last few years, you might recognize this as very similar to the structure that they animate for COVID-19. So I'm going to talk about this with respect to HIV, and then I'll briefly touch on COVID-19. Essentially, the HIV virus has these little receptor markers on the outside of their cell. Those have evolved to recognize something called a CD4 marker on the cells of our immune system. So viruses cannot infect just any old cell. They can only infect cells that they can recognize. And the HIV virus recognizes these CD4 markers. These markers are on specific immune cells within our body. When they recognize and bind to those markers, they can be pulled into the cell, and the virus essentially takes over the cell's machinery, starts producing more viruses. If a cell does not have CD4 markers, it won't infect it. So this is why HIV is not spread like through respiratory droplets. You can't sneeze on somebody and give them HIV because one, they're not sneezing out virus particles. And two, even if they were, they wouldn't be attaching to the cells in your respiratory system because they don't have those CD4 markers. COVID-19 though does have that crazy spike protein that we keep hearing about, and that does recognize our respiratory cells. They recognize something called an ACE receptor on our respiratory cells. That's why if you breathe on somebody while you are infected with COVID-19, you release virus particles. They land on the nose, throat, etc. of whoever you're breathing on, and they can attach to the ACE receptors in the cells that line your respiratory structures. And that is how the virus enters those cells. So the final membrane function that we're going to talk about for the plasma membrane that makes it so important is the transport function. Remember, things need to come in and out. Oxygen in, carbon dioxide out, etc. So there are several ways that molecules can enter a cell. Before we get into those particular transport methods, I want to talk about something called diffusion. Now, even if you've not taken a science class before, you probably have an idea of what diffusion means. Diffuse means to spread out or to dissipate, right? You've probably talked about diffusing the situation, spreading out the situation, calming it down, that sort of thing, relaxing the tensions, right? So diffusion is actually the spreading of something out. So one of the ways that things enter and exit the cell is by diffusion. But diffusion does not have to cross a membrane for that to happen. Diffusion is simply spreading out from an area of high concentration to an area of low concentration. So imagine a glass or bowl of water and you drop some food coloring in it. Or imagine a cup of black coffee and you pour a little bit of creamer in it. What happens? It spreads out. That drop is an area of high concentration. And if you don't stir it, we're not talking about stirring right now. If you don't stir it, those molecules that are in really high concentration are actually on their own going to sort of bounce against each other and eventually spread out. They are diffusing away from the point of high concentration. So those creamer molecules or food coloring molecules are going to spread out from their high concentration to areas of lower concentration until they eventually reach equilibrium, where they're all evenly spaced. So diffusion is any movement from high concentration to low concentration. Now, this is a passive event. It's going to happen without any input of energy, without any outside forces. So cells provide cell membranes allow for passive transport in two different ways. One is what we would call simple diffusion. It is where molecules move from an area of high concentration straight through the membrane to areas of low concentration. So here we have an example of oxygen molecules, which are small and non-polar. So because they are small, they can move directly through the plasma membrane, which a few minutes ago I said wasn't really possible. It actually is. If the molecule is small enough and has the right properties, it actually is. So oxygen is really small and it's nonpolar. So oxygen actually moves by diffusion across the plasma membrane. Carbon dioxide is also small and nonpolar. Oh goodness, nonpolar. So it can also diffuse through from areas of high concentration inside to areas of low concentration outside. So that's simple diffusion. This doesn't require any energy. It doesn't require any action on the part of the cell. Think of what the word passive means. When you let something happen around you, you're not participating, you're not making it stop, you're not making it go. That's passive. It just happens on its own. Another form of passive transport is what we call facilitated diffusion. So in facilitated diffusion, we still have diffusion working. We have an area of molecules that are in high concentration moving to low concentration. But with facilitated diffusion, we have molecules that are too big or generally polar, or they can be nonpolar too. Too big, but generally polar, and cannot just cross the membrane on its own. In order to get into the cell, even though it's going by diffusion, it requires a transport protein. So this is a big protein with a channel down the middle. So it's a channel protein that allows large molecules to move from areas of high concentration into the cell where they're in low concentration. So the principle of diffusion is still happening here, still moving from areas of high concentration to low, but this time we have a protein that is facilitating that. It's actually helping this to happen, and that is because it has a channel through which those big polar molecules can move freely.